So we're going to talk about some reactions that are central to life on Earth, namely photosynthesis and cellular respiration. So this is how organisms get and use energy for all the things that living organisms need, like growth and maintenance, reproduction, defense, manufacture of all these chemical compounds, all these reactions going on, all the energy that's needed for life. It turns out, if you remember back in, we talked about chapter 3, chemistry, the first law of thermodynamics is that, namely, energy and matter can neither be created nor destroyed. So, if you remember when we looked at reactions, you have to have the same number of atoms of a certain element on one side of a chemical reaction as another. So let's say we have methane on one side, C, and then H4 plus H2O. And then you have the reaction. On the other side, you have to have the same number, so there has to be a carbon over here. So carbon monoxide, and then 6 plus 3 H2, so that's 6. So 6 hydrogens on one side, 6 on the other, a carbon and a oxygen stuck together, carbon monoxide. Okay. So chemical reactions, all the things that cells do are involved in are called metabolism. That means making things, breaking things down, all these chemical reactions, and they happen inside a cell, because these things happening outside of a cell would be too difficult. And these metabolic reactions happen in metabolic pathways, where many times a bunch of different reactions and things happening are all strung together. So, in other words, the key biological molecules, amino acids, can be strung together to make proteins. Nucleotides, nucleic acids, can be strung together or read to make DNA, or to be read and turned into an RNA. You can make more complex fatty acids for plasma membrane and all that, or make long-chain carbohydrates using glucose. And you can break down all these molecules as well. So these are what metabolic pathways do. So when metabolic pathways, when a reaction happens that takes simple things and makes them into more complex molecules, that's called anabolism. That create complex molecules from simpler ones, namely, like I showed here, from monomers are strung together using energy into polymers. Okay, and then you can also go the other way, take polymers and break them down into simpler monomers. That's called catabolism or catabolic. Often catabolic, usually enzymes do both of these different things. They can either add things or break things down. So, I went on from this. So enzymes. Okay. So enzymes are proteins. In other words, they're coded for in the DNA. That DNA is transcribed into RNA. That RNA goes out of the nucleus into the cytoplasm. And the ER combines with ribosomes, makes proteins. And some of those proteins are enzymes that allow reactions to happen in a more efficient way and a safer way inside of a cell. Okay, so let me explain. In chapter 3, we talked about reactions. Some reactions require energy to happen, others don't. So let's say a good example is when you light wood and it starts to burn, there's a chemical reaction happening. It's actually you're breaking all these cellulose bonds, which are carbohydrates. That releases a lot of heat, and you end up with carbon. The thing is, you have to heat up the wood to a very high temperature. So this is the level of the reaction right here, where you need to put so much energy into the reaction before the reaction then will carry on its own, and the wood will burn up until there's nothing left. So let's say here's the wood at the very bottom and beginning, and it has to climb all the way up to the reaction energy before the rest of the reaction can happen as the wood burns, and then you release more and more energy until there's no energy left. It's been released into the atmosphere, and you've got carbon. Okay. The thing is, if you did that inside of a cell, it would burn up a cell, because we're talking very 300 or 400 or 500 degrees Celsius in terms of burning something like carbohydrates up. So enzymes allow us to actually do these reactions, but instead of doing them all at once and needing a lot of energy, you do it all in small steps. So let me show you something. So let's say we've got a mountain, and one way to get up it is to climb it. So here's a person, and that person has to climb it all up. It's very difficult. It's dangerous. You have to use ropes and all that. They expend a lot of energy, of course. Fine. Hopefully they get up to the top and they've got a nice view. However, you can also climb this mountain a little bit easier. So instead of going straight up, you can take a circuitous route and you do a bunch of switchbacks, a road, all the way up until the top. It takes a lot longer. So from here to here might be 1,000 feet. Whereas from here might be 5,000 feet. Okay? The thing is, you're going to use the same amount of energy to get up, but it's going to be a lot easier to get up with the switchbacks. Okay? So it turns out that enzymes do the same thing. So this is equivalent right here to climbing the mountain directly, whereas this right here is equivalent to doing it more slowly. And what it does is, instead of having to go all the way up here, all that energy right away, you actually make it so that you can only need a certain lower amount of energy, but you make it in a whole bunch of little steps like I showed here, right? So that eventually you end up with the same product, but you don't it can happen at a lower temperature, and it can be more controlled, and it makes it possible for things to actually, reactions that couldn't happen inside of a cell actually happen inside of a cell. So that's one thing that enzymes do. They catalyze reactions, usually by lowering what's called the activation energy that I mentioned earlier. Okay. So here's an enzyme. Usually proteins have a three-dimensional structure. Enzymes certainly do. And enzymes, and it does this with what's called induced fit. As molecules bind to the active site, the enzyme changes shape. And then once it releases, it lets it go. So the enzyme is not permanently changed by the reaction. It can be used over and over again. Actually go next over here. Okay, so here's our enzyme. It's a protein, right? And then it's got what's called an active site. And then if we look at this, we have let's say it's going to put two molecules together into a third molecule, a bigger molecule. So it's going to do this because it has an active site where the two different molecules come together right here, and they're put together in a new molecule. Okay? And it changes the actual shape of the enzyme when that happens. The product, these two molecules now into one, is released, and then it can go and do its work. And then the induced fit, the molecule with the active site, it changes shape, and once it's released and goes back to its original shape, it can take in more molecules and put them back together, the same kinds of molecules. So why it's important that enzymes not be permanently altered when they bind to substrate molecules? Well, they can be used over and over again. And again, a lot of proteins can be used over and over again because you can always break them down. And proteins are made of amino acids, and you can break them down into simpler amino acids that can be used again and again to make new proteins. So a good example of this is lactose. A lot of people cannot drink milk. Some people cannot drink milk as adults, but some can. So lactose is, among other things, it's a sugar. It's made up of a glucose molecule and something called the galactose molecule. And if you have the right enzyme called lactase, you can break those apart and use those sugars in cellular respiration. And so in milk, in addition to other things that are in it like fats and things like that and other proteins, it has a lot of sugars that can be broken down, but you need the enzyme for it to happen. Normally with mammals that make milk, female makes milk, it takes a lot of energy to actually make milk. And of course, at a certain point, the mother weans, tells the offspring to go get lost and find your own food. Okay? So during that time, there's a system a gene that's switched on with a promoter region that's upstream of the actual lactase gene that codes for the enzyme. And it's making lactase all the time. The baby's drinking milk, it gets broken down, can use all that energy. But of course, once the baby is weaned and it's off on its own, it's eating regular food, it's not going to come across milk anymore. And making an enzyme you don't need is very expensive. So normally what happens is that promoter region then gets changed and shut off, and that lactase enzyme is no longer producing lactase because mammals never come across milk anymore. Well, in humans, there was a mutation in a number of different populations independently. And at a certain point, humans domesticated animals, and mostly what they domesticated for food were ungulates like sheep and goats and cattle and things like that. And originally, those cattle, of course, and sheep and goats make milk, but they really couldn't drink it because they didn't have, as adults, babies could drink it, but adults couldn't because they weren't making lactase. However, it turns out that a lot of humans were still using milk, but they were changing it. They were using fermentation, a different process, to turn it into cheese, which then turns it into a combination of fat and glucose that can be broken down. And so these dairying herders would run their livestock around to various areas for food over the course of the season. They'd, of course, slaughter some of the animals. They'd use hides, and they would also use the milk, but to make it into cheese that they could use for food. Okay. At one point, in a number of different populations, there was a mutation in an individual. And that individual, the mutation did was, in the promoter region, it kept the lactase gene producing lactase, the protein for breaking down milk. If there wasn't milk to be available and you're still producing that, that's a bad thing. But it turns out here's an environment where there's milk, and this individual, instead of having to wait to turn it into cheese, they could drink milk directly. This gave them an advantage in terms of a food source. They could drink milk all the time. They didn't have to go out hunting. Let's say the animals were producing milk, but there wasn't much else to eat. They could still survive, and in doing so, they would reproduce, and others might not. So eventually, this mutation spread through the population. More of that individual's descendants, offspring, grandkids, great-grandkids, all that, might have inherited this gene, and as long as there was milk, they were getting a benefit compared to everybody else. So that's an example of what an enzyme. So glucose itself doesn't do much good, but it's a high energy molecule, but you can transform it through a bunch of chemical reactions into ATP. And ATP powers all the activities in all the cells. That's the energy. If you don't have ATP, nothing works in the cells. If it doesn't have ATP, it'll die. And then there's these other molecules that are related, that are high energy molecules, that some are higher energy than others, and they get broken down. Okay, so I mentioned that reactions are often strung together in terms of metabolic reactions. And different metabolic reactions that are strung together require energy, and along each part of it, either energy is added or energy is released. And the molecule that's used, as I mentioned, is ATP, adenosine triphosphate. It's got adenosine. I could show you what it looks like, but forget about it. And then there's 3 phosphoruses attached to it. And when you break down ATP, you usually first lose a phosphate, and that releases energy that can be used to drive reactions. So ATP, usually it's made from glucose or some other carbohydrate, and every cell needs ATP. So all cells in the body and all cells outside of the body and bacteria and everything else, they actually need ATP to power cell respiration. If the cell doesn't have ATP, it dies. ATP then can also, it can transfer some of its energy into other molecules that are energy carriers that we'll take a look at. So, cells store and deliver energy using ATP. Here's the ATP. You see adenosine. It's a chemical, and 3 phosphates. When you break up a phosphate, one of them, you release energy. If you break up another one, you release more energy. Now you've got ADP, which is diphosphate. Now, if you add energy to this reaction in the form of sunlight, you can actually make an anabolic reaction by adding a phosphate to the group. In other words, to this group. Okay? So, but you need an outside source. Okay. So, ATP is made from glucose. It's here's ATP. The energy is adenosine, and then it's got these 3 phosphates. If it's part of a reaction with an enzyme that cleaves off a phosphate, energy is released for the cell that can be used. Okay? It turns out when you do that, then you end up, instead of having ATP, you have ADP. It's a lower energy molecule. You can also lop off another molecule, or in fact, you can add phosphates back onto it during photosynthesis. Okay? So this is kind of how, this is a circular reaction. Here you're adding an anabolic reaction, here catabolic, breaking off, okay, and releasing energy. Okay, glucose metabolism. Almost all life on Earth uses glucose. What organisms make glucose from the ground up? Anybody? What do you think? Yeah. Think about what makes wood. Wood. You know, wooden trees and all that. What makes that? What kind of organism? Plants. Okay, good. So, some organisms actually make their own products and their own energy and make glucose. And those are plants, some bacteria, some different kinds of algae. They're called autotrophs. They make all the glucose that the rest of the planet runs on. Everything else eats what the autotrophs make, and they're heterotrophs like ourselves. So there's 2 chemical reactions going on here. The autotrophs have an anabolic reaction, and the heterotrophs have a catabolic reaction. They break that down. So I'll leave you with that, but this is the whole reason that if you combine these 2 systems together, the whole thing works. Okay. So, in terms of glucose metabolism, some organisms are able to make glucose. There's some bacteria called cyanobacteria that are actually the basis of chloroplasts in algae and in plants that can take sunlight and transform it carbon from CO2 and then water and turn it into glucose, which is a higher energy molecule. It's a sugar, and then you can tie a whole bunch of those together. So they actually make glucose. Only about 5% of organisms actually can do that, like plants, some other algal species, and cyanobacteria. Everything else has to get their glucose that they then break down to produce ATP. They have to get that by eating organisms that actually produce glucose, namely cyanobacteria, algae, a number of other things that plants that actually produce glucose. Okay, so the ones that produce are called autotrophs, and they create carbs and carbohydrates, and these are plants, and they do this with photosynthesis. And then heterotrophs take those carbohydrates and they break those down and turn them into simpler sugars that are then turned into ATP. And then are also used to make fats and proteins and things like that. Okay, this is as far as we went.